Hello everyone. In this class, we'll learn about neural regulation of respiration. Spontaneous respiration is produced by rhythmic discharge of motor neurons that innervate the respiratory muscles. This discharge is totally dependent on nerve impulses from brain. During regulation of respiration, pulmonary ventilation is adjusted according to the metabolic demands of the body. The metabolic demands are the supply of oxygen to the tissues, removal of carbon dioxide during metabolism, and maintenance of optimum pH of the blood. Unlike all other organ systems, respiration demonstrates automaticity as well as self-modulation. That is, we breathe without thinking, but we can willingly modify the breathing pattern. Rate, depth, rhythm of respiration is controlled by a group of neurons situated in reticular formation of brainstem. The collection of these neurons are called as respiratory centers. The rhythmic discharge from the brain that produces spontaneous respiration is regulated by two mechanisms, that is nervous regulatory mechanism and chemical regulatory mechanisms. Respiratory centers. The rhythmic discharge is initiated by a group of pacemaker cells in the brainstem referred as pre-Botzinger complex of neurons, PBZ. They are situated in either sides of medulla. These neurons discharge spontaneously and rhythmically. They produce rhythmic discharges in phrenic motor neurons. All the neurons regulating respiration project into PBZ. The respiratory centers are situated in the reticular formation of the brainstem, and depending upon the situation in the brainstem, they are classified into medullary and pontine center. There are two centers in each group. Medullary centers include two groups of neurons, the dorsal respiratory group of neurons, that is DRG, and ventral respiratory group of neurons, that is VRG, which generate basic respiratory rhythm. The pontine centers include apneustic center APN and pneumotaxic center, that is PNC, both of which modify the activity of medullary respiratory center. Pontomedullary respiratory center neurons are of two types, that is I and E neurons, that is inspiratory and expiratory neurons. I neurons are active during inspiration and E neurons are active only during forceful expiration. This diagram showing all the uh, pontine and medullary respiratory centers. So these are the medullary respiratory center. Those are dorsal respiratory group of neurons and ventral group of neurons. And in the pons, we have two groups, pneumotaxic center and apneustic center. And the important nuclei here is the dorsal respiratory group of neurons, which are influenced by uh, the pre-Botzinger complexes. The impulses which are produced in the pre-Botzinger complex, they reaches the dorsal respiratory group of neurons, and they are going to affect the anterior motor neurons, which are supplying the diaphragm and respiratory muscles. And amongst these centers, the dorsal respiratory group of neurons plays a very important role, and in turn they are regulated by the ventral respiratory neurons and from the pons by the apneustic center and pneumotaxic center. Pneumotaxic center, it tries to always inhibit the apneustic center, and apneustic center, it stimulates the inspiratory group of neurons. Okay, and if there is a lesion between the pneumotaxic center and the apneustic center with a vagal denervation, it is going to cause apneusis, that is the respiration will stops in uh, inspiratory phase, so which is called as apneusis. Dorsal respiratory group of neurons. Most of the neurons are located within the nucleus tractus solitarius, and some in the adjacent reticular substance. The basic rhythm of respiration is generated mainly in the DRG. They contain mainly the eye neurons. The DRG send fibers to the phrenic motor neurons which innervate diaphragm. The nucleus tractus solitarius is the sensory termination of both vagal and glossopharyngeal nerve which transmits sensory signals to the respiratory centers from peripheral baroreceptors, chemoreceptors, and pulmonary receptors. Inspiratory ramp signal in the inspiratory ramp signal, the nervous signal that is transmitted to the inspiratory muscles, mainly diaphragm, is not an instantaneous burst of action potential. It begins weakly and it increases steadily in a ramp manner for about 2 seconds. Then it ceases abruptly for about next 3 seconds. During this period, expiration occurs. At the end of third second, the inspiratory ramp signals appear in the same pattern and the cycle is repeated. So this is the pattern, like inspiratory signals gradually they increase for the first 2 seconds and abruptly stops. Okay. And then for next 3 seconds there will be expiration. Again the next cycle is repeated. That is gradual increase in the inspiratory ramp, then suddenly stop within 2 seconds. So like this, the cycle is repeated. Ventral respiratory group neurons. Ventral group is long column of neurons that extends through nucleus ambiguous and nucleus retroambiguous in ventrolateral medulla. They contain both inspiratory and expiratory neurons. Inspiratory neurons are situated in its mid portion, whereas the E neurons, expiratory neurons, at the caudal and rostral end. Normally, this center is inactive during quiet breathing and it becomes active during forced breathing or when the inspiratory center is inhibited. The rhythmic discharge of neurons in the medullary respiratory center is spontaneous, but it is modified by the neurons in the pons and by afferents in the vagus nerve from the receptors in the airways and the lungs. So these structures are going to receive the negative feedback mechanism. Pneumotaxic center situated in the dorsal part of the upper pons in nucleus parabrachialis and caulico-fused nuclei. It contains both inspiratory and expiratory neurons. The main function of the pneumotaxic center is to switch off inspiration so that the duration of inspiration is controlled. Indirectly, the pneumotaxic center increases the respiratory rate by limiting the duration of inspiration. That is, when the duration for inspiration is reduced, naturally the expiration time is also reduced so that respiratory rate increases. Apneustic center situated in the reticular formation of the lower pons, it is made up of diffusely located neurons in the region of nucleus pontocaudalis and rostral part of the nucleus gigantocellularis. This center is always excitatory to medullary inspiratory center and it increases the depth of inspiration. Neural regulation, that is the two separate mechanisms, are the voluntary control and automatic control. When you come to the voluntary control, normally the respiration is involuntary in action. Voluntary control is possible, but only to a limited extent. Breathing can be voluntarily inhibited only for a short period of time. It becomes voluntary in spite of all our efforts to hold our breath. The point at which breathing can no longer be inhibited is called as breaking point. The duration of the breaking point is around 40 to 60 seconds. The breaking occurs due to the rise in partial pressure of carbon dioxide and fall in partial pressure of oxygen. Breaking point can be delayed by removing carotid body, breathing 100% of oxygen before breath holding, and hyperventilation. Breathing, break, breathing point is reduced in exercise or carbon dioxide rich in air. Breaking point. Center of pathway for voluntary control is cerebral cortex. Impulses are sent to the respiratory motor neuron via corticospinal tract. These nervous pathways bypass the medullary respiratory center neurons. As automatic control is through neurons of pons and medulla, pathway for voluntary and automatic control are separate. Automatic control of respiration is sometimes destructed without loss of voluntary control, and this clinical condition is called as Ondine curse. This is seen in patients with poliomyelitis. Automatic control. Automatic control of respiration is mainly through respiratory centers. The nerve fibers from respiratory centers leave brainstem and descend in the anterior part of the lateral column of spinal cord. These nerve fibers terminate in the motor neurons in the anterior horn cells of the cervical and thoracic segment of spinal cord. From the motor neuron of the spinal cord, two sets of nerve fibers arise, which are phrenic nerve fiber that supplies the diaphragm and the intercostal nerves which supply intercostal muscles. Impulses from higher centers and various reflexes influence respiratory centers. Control from higher centers like respiration is modified during deglutination, chewing, speaking, crying, and singing. This modification is done with the help of cerebral cortex, hypothalamus, and limbic system. Reflex control. Then Herring-Brewer reflex. The impulses from lungs bring about respiratory reflex called Herring-Brewer reflex. This reflex prevents over distension of lungs during inspiration and collapse of lungs during expiration. This is not important in the normal adults in the range of normal tidal breathing, and it is important in neonates and infants. There are some stretch receptor on the muscular portion of the wall of the bronchi and bronchioles of the lungs. Overstretching of the lungs during inspiration stimulate the stretch receptors and impulses are carried by vagal afferent fibers to DRG, dorsal respiratory group of neurons, and inspiration stops and expiration starts. This reflex is a protective reflex because it restricts the inspiration and limits the overstretching of the lungs. This is called as Herring-Brewer inflation reflex. However, this reflex does not operate during quiet breathing. It operates only when the tidal volume reaches above 1500 ml. The reverse of this reflex is called as Herring-Brewer deflation reflex. When the inspiratory center is inhibited, the inspiration stops and expiration occurs. During expiration, as the stretching of the lungs is abolished, the deflation of the lung occurs. J receptor mediated reflex. J receptors are juxtacapillary receptors. These receptors are the sensory nerve endings of the vagus. The fibers from these receptors are non-myelinated and belong to C type. The J receptors are situated in the alveolar interstitium close to the pulmonary capillaries. J receptors are stimulated by certain chemicals released in the interstitium during pulmonary edema, pulmonary embolism, and pulmonary congestion. Stimulation of J receptors produces reflex response, which is characterized by a period of apnea followed by rapid breathing, bradycardia, hypotension, and weakness of skeletal muscles. J receptors may have a physiological role in severe exercise. Severe exercise, muscular exercise increases the mild degree of collection of the fluid in the alveoli, which causes irritation of the J receptor, which leads to muscle weakness and the person stops exercising. That is, the breaking of exercise occurs. Lung irritant reflex. It is also protective reflex. Receptors are located between epithelial cells of tracheobronchial tree The stimulus is exposure to irritant fumes or dust, and stimulation of irritant receptors produces reflex bronchospasm manifested as a cough and sneeze, so that irritant fumes cannot reach to the alveoli. Then reflexes from the muscle spindle of diaphragm and the other inspiratory muscles. In case of insufficient air entry in the lungs, as in case of airway obstruction, the muscle spindle act in such a way that inspiratory muscles continue to contract more forcefully, and this will help to overcome the obstruction. Reflexes from the limbs and joints. Movements of limbs causes stimulation of proprioceptors, that is muscle spindle and Golgi tendon organ, and this leads to increasing the rate and depth of respiration, and the reflex is very active during exercise. Baroreceptor reflex, that is rise in the blood pressure, arterial baroreceptors are stimulated and afferent impulses terminate on the vasomotor center and respiratory center. Particularly the inspiratory neurons are inhibited, which leads to stoppage of respiration or inspiration. Then we have a chemoreceptor reflex, and that we are going to discuss. And cough reflex is a protective reflex. Cough begins with a deep inspiration followed by forced expiration against closed glottis. This increases the intrapleural pressure above 100 mm of Hg. The glottis is suddenly opened, producing an explosive outflow of air. Then sneezing reflex, it is similar to expiratory effort with a continuously open glottis. Then we have deglutition reflex. During swallowing of the food, there is a temporary arrest of respiration, is called as deglutition apnea, and this occurs during the pharyngeal stage, that is the second stage of deglutition, and it prevents the entry of food particles into respiratory tract. The nerve involved in this reflex is glossopharyngeal nerve. Others like rise in the body temperature causes hyperventilation, and sharp acute pain increases the rate of respiration temporarily. This is in brief about the nervous regulation of respiration. Thank you.